So just a bit of intro. So I am Professor of Epigenomics at the University of Exeter Medical School. So my lab is really interested in understanding both causes and consequences of molecular variation in the brain. And we're interested in that in the context of a whole range of neuropsychiatric and neurodegenerative diseases. So I thought I'd just start by putting this in a bit of context. You've heard about one sort of brain disease in terms of pediatric brain cancer in the first talk today. So we're focused on non-malignant diseases, but this is data from the World Health Organization, just highlighting that these neuropsychiatric and neurodegenerative disorders make up almost about 30% of non-communicable disease in the developed world. And just to put a few stats out there, almost 47 million people in the world are currently living with Alzheimer's disease. It's estimated that by the end of this year, 1 million people in the UK will have some form of dementia. And although you'll have all heard about some of the kind of recent successes in perhaps developing new disease-modulating therapies, actually we're still really a very long way from realizing the potential of drugs to treat these disorders. So with the aging population in the UK and elsewhere, we're really kind of setting ourselves up for a demographic time bomb with many of these phenotypes. So just like any other type of disease that has a high inherited or genetic component, advances in our ability to genotype and sequence populations at scale mean that we've made huge advances in identifying genetic risk factors for pretty much any complex brain phenotype. And epidemiology has pointed us in the direction of many risk factors, but how these things come together to mediate risk in disease-relevant cell types is still, very much an unknown. And the kind of real key challenge, I think, facing us now we have these genetic variants associated with disease, is to really understand the functional consequences of these genetic risk variants. And of course, by doing this, it will allow us to think about mechanisms in disease, which will allow us to point ourselves towards new drug targets, biomarkers that can maybe be used to stratify individuals and help develop the treatments that are going to really help us long term. So as I said, the work in my group really does two things. It's first focused at really annotating how genes are regulated in both the developing and aging human brain, and I'll talk about some of that work later. And the second component of our work is then in looking at individuals who have died with a disease to see if we can understand or identify molecular signatures of the disease process in the relevant tissue or cell type, and use that to make inferences about mechanisms in disease. And we do this by profiling many layers of regulatory genomic variation using a whole range of different technologies, and I'll talk about some of those today. So just as we can now genotype individuals and sequence the DNA from individuals at scale, we now have the technologies that mean we can profile modifications, epigenetic modifications at scale. And you heard about DNA methylation in the previous talk, and we can now profile DNA methylation in brain tissue from autopsy brain donations from individuals who've died with and without disease. And this approach has been really, really successful. So it's allowed us to identify regions in the genome that are epigenetically modified in the brain in individuals with diseases such as Alzheimer's disease. And this has really highlighted some novel biology. It's given us insight into mechanisms, and as I've alluded to, has sort of started pointing us towards potential for novel drug targets. Just to give you an example, this is just, so as I said, we're focused on DNA methylation. This is a study of 1,000 individual brain samples, and you can see the different distribution in DNA methylation levels at a site in the promoter of the ANK1 gene. in individuals who've died with Alzheimer's disease compared to those who have no Alzheimer's disease in their pathology. What you'll notice here is that there is clearly a different pattern in those two groups, but actually the shift in that pattern is really, really small. And what this tells us is that we need to do much bigger studies, just as genetics have, in order to identify small effect sizes. But it also tells us that maybe some of the signal that we're getting from profiling the tissues we use is maybe confounded by various factors, which I'll talk about in a minute. The things to note are the signal that we see is specific to the region of the brain that's affected in Alzheimer's disease, so that's the entorhinal cortex. We don't see it in regions of the brain that aren't affected by the disease, so regions like the cerebellum that you don't see any neuropathology in Alzheimer's disease, and certainly not in pre-mortem blood samples taken from the same donors. So this tells us straight away that it's very important to be looking at disease-relevant tissues when trying to identify the molecular signatures of the disease. So we've built on these initial results. This is a recently published study from our group that's now profiled over 2,000 brain samples. We think this is probably the biggest study like this on Alzheimer's disease yet performed. We've identified over 300 regions in the genome, which are annotated to just over 170 genes that show highly significant patterns of differential DNA methylation associated with Alzheimer's disease pathology. And just to note that these list of genes are really enriched for immune and neuroinflammatory pathways, and that's very interesting because that really highlights an aspect of biology that we know is important in the onset of Alzheimer's disease pathology. So one of the key limitations of all these types of work is that they're generally performed on lumps, bulk lumps of cortex tissue that have been taken from individuals at autopsy. And as you can see from this image here, the brain is obviously a really heterogeneous mix of different cell types. And each one of those cell types will have a very specific epigenetic signature. So there's a lot of cellular heterogeneity going on. And of course, in Alzheimer's disease, that's really confounded because the disease itself is really associated with a loss of a very specific cell type in the brain, the neurons, and it's also associated with the proliferation and activation of another cell type, the microglia, the kind of immune cells of the brain. And so there's kind of real potential for confounding in these studies that are performed on lumps of tissue. So to combat this, we have developed methods to enable us to isolate DNA and chromatin from purified populations of nuclei from the different cell types of the human cortex. We essentially label the nuclei with markers that are specific for the different cell types, put them through a fax machine, and then we can collect enough nuclei to enable us to do downstream processing in terms of many different types of multi-omic profiling. So it's a really efficient approach. If people are interested in the method, you can scan the QR codes. We validated that the populations enriched are really what they say they are by using single-cell RNA-seq. And just to give you an insight as to why that is a really powerful approach. So what you can see here on these series of correlation scatter charts, each dot represents one of those 330 sites that we found differentially methylated in Alzheimer's disease brain when we were looking at the bulk cortex tissue, and you can see the effect size there on the x-axis. On the y-axis is the effect size at the exact same position in the genome in purified populations of neurons on the left, oligodendrocytes in the middle, and those microglia cells that I told you about earlier on the right. And what you'll see here, just by looking at the scale, is that the signal is really all coming from these microglia cells. So this really highlights the power of looking at epigenetic changes in very specific cell types. And we're now doing this for all the diseases and phenotypes that we study in the brain. So while the signal in Alzheimer's disease is all coming from microglia, the signal in schizophrenia is coming from a different cell type in the brain, specifically excitatory neurons. And you can see here the network of genes that we find differentially methylated in the brains of individuals who've died with schizophrenia, which is a very severe psychiatric phenotype. Now, I've put this up because what it highlights is two things. So firstly, that these sites of differential methylation are coordinated. They kind of move together in these networks of differential DNA methylation. And these networks are highly enriched for neurodevelopmental pathways. And that's really interesting because it's been strongly hypothesized that schizophrenia and other neuropsychiatric phenotypes have their origins during brain development. And actually very little is known about the epigenetic signatures that change across development of the human cortex. And so this set us on the path to really sort of fill in that gap and generate maps of gene regulation across the developing and then the aging brain, with the idea that if we can understand what changes are taking place in normal development and aging, that will allow us to understand more about what's going on in the context of disease. So the cartoon here just shows you the ages of the samples that we've profiled in this recent study. So they range from about 7 weeks post-conception right up to just under 100 years of age, and they've been profiled for DNA modifications, histone modifications, and various transcriptional readouts. And I won't go into huge detail about the results, other than to say that we see really dramatic changes in gene regulation across that prenatal period. So in these scatter plots on the bottom right, these are just two sites. You can see one site that's becoming dramatically hypermethylated across brain development. On the right, a site that's becoming very rapidly demethylated across brain development. And these changes here, each of these bars represents a week in terms of gestational age, and essentially these sites are changing totally from being unmethylated to methylated and methylated to unmethylated. Again, these studies have all been done on purified cell types, and you can see here that the changes are very cell type specific across this period. So again, this dotted line here represents birth. Anything on the left is prenatal, anything on the right is postnatal. And in green, you have the non-neuronal cell types, and in pink are the neurons. And you can see very specific changes in the epigenetic state of those genes across the developmental and postnatal period. So we were interested to see if the genes that are changing across development are enriched for the same genes that are have been highlighted from genetic studies of neurodevelopmental phenotypes. And what you can see in each of these bar charts here are the gene sets from studies of both autism and schizophrenia, these disorders that are hypothesized to have neurodevelopmental origins. And the bottom line is that we see very highly significant enrichment for genes that are being epigenetically altered across brain development, overlapping with those genes that are enriched for genetic variants and mutations in the onset of those disorders. So this, I think, highlights one of the power of generating these type of regulatory annotations, because it allows us to put together what we've learned from genetic studies of disease with what we're learning from profiling the epigenome and transcriptome in different cell types at different stages in life, and allowing us to really home in on potentially the function of the genetic variants associated with disease. It's worth noting that most of the variants that have been highlighted by genome-wide association studies don't alter protein structure. They're hypothesized to affect the expression of genes. And actually, the genetic variants that are highlighted by GWAS are often very non-specific because of the nature of linkage disequilibrium across the genome. It's often very hard to prioritize the actual causal gene. And so by generating these functional maps, it really allows us to start homing in on disease-relevant regions of the genome. just to highlight that, so this is work by Eilish Hannon in my group, who just basically essentially showed that the genetic variants associated with pretty much any brain disorder are highly enriched in regions of the genome that are known to regulate gene expression. So in this case, we're looking at H3K27 acetylation peaks in neurons. These are regions of the genome that act as active enhancers or promoters for gene expression. And the genetic component, the polygenetic component for these disorders from GWAS is highly enriched in these regulatory regions. And what's interesting is that when you look across the range of all GWAS studies, that enrichment is really very specific for the CNS or brain traits. In fact, we don't really see any enrichment at all for GWAS results from traits that don't necessarily have, or that don't look like they have anything to do with the brain. Again, this highlights the importance of generating these reference annotations in disease-relevant tissues and cell types. The other thing that we know is that inter-individual variation in the epigenome, although I've highlighted, it's very cell type specific and it changes dramatically over development, the variation between individuals is under very strong genetic control. So this is a study looking at genome-wide DNA methylation in 2,000 pairs of monozygotic and dizygotic twins. And because obviously MZ twins share all their genetic variation in common, and dizygotic twins share on average half of their genetic variation in common, it allows us to work out the extent to which methylation at any given site in the genome is under genetic or non-genetic control. What you'll see here is a very sort of strong shift towards MZ twins being more similar to each other than DZ twins, highlighting a genetic component to overall levels of DNA methylation. And here's the data for just one site in the genome. Each dot here is an individual twin pair, twin one on the x-axis, twin one on the right. This column here is for monozygotic twins, and here's dizygotic twins. And what you'll see is a much stronger correlation in the MZ twins and the DZ twins, telling us that at this particular site, genetics plays a role in the level of methylation. And then we can actually map genetic variants that actually impact very strongly on methylation at that site. And these are what are called DNA methylation quantitative trait loci, or MQTLs. It's interesting to me that there are sites that show high levels of variation that aren't under genetic control, so that the level of variation is the same in MZ twins as DZ twins, and these are sites that may well be under environmental control, because we know that aspects of the environment can also impact upon levels of DNA methylation and other epigenetic marks. So we can use the fact that we have these samples with whole genome methylation profiles. We can sequence or genotype those individuals across the genome and start mapping the genetic variants that impact upon DNA methylation at any given site. And so we were very interested to generate these MQTL maps in our developing brain samples. And what you can see here is that very much the genetic effect on DNA methylation at any site can be very developmental specific. So this is one site in the genome, and the three bars depicts the three different genotype groups, and then you've got different, you've got the fetal cortex here in red, the adult cortex in blue, another region of the brain, the striatum in green, and the cerebellum in orange. And what you'll see here is that the genetic effects on DNA methylation here are both region specific in terms of what region of the brain you look at, but they're also developmental specific. And in fact, the same genetic variant can have opposite effects on DNA methylation between the fetal cortex and the adult cerebellum. Again, it highlights the real importance of generating these MQTL maps, these modification maps in cell types and developmental periods that are relevant to the study, disease that you're interested in studying. And just to show you the utility of this, what we're able to do is then take the GWAS of schizophrenia and show that it was highly enriched for these methylation QTLs that are specific to the fetal cortex. Again, reinforcing the fact that the genetic component to schizophrenia is likely operating through genes that are being regulated differentially across brain development. And these molecular QTL maps have real utility downstream in fine mapping regions of the genome that have been highlighted from genome-wide association studies. So one of the outputs of a GWAS might be highlighting a region like this that might contain 6, 7, or 8 genes, but by using the co-localization of genetic and epigenetic and other regulatory marks in that region, we can start focusing on specific genes that then can be the focus of downstream functional characterization works. So one question we often get is, well, so what? If you find these epigenetic variants, what does that mean in the context of gene expression? And an area that we're particularly interested in is the effect that these epigenetic marks, particularly those that are dynamic across development, might have on transcription at the level of individual transcripts, and particularly in relation to isoform abundance and alternative splicing. So just as a little bit of an intro, so we know that most of our genes in our genome undergo some sort of alternative splicing. It's a real important mechanism in the developmental control of gene expression. It's particularly important in the central nervous system. And the reason it's important to look at is that the different transcripts from the exact same gene can have very different functional effects, sometimes totally antagonistic effects in terms of their function. One of the limitations of standard gene expression analysis based on short read sequencing is it's very hard to look at the actual structure of the RNA molecule that's being expressed. So we've been using long read transcript sequencing, both the ONT and PacBio approaches, to sequence entire transcripts that allows us to unambiguously look at transcripts and detect the different isoforms that are detected in the cell types that we're looking at. And just to give you a cartoon of, again, what this is allowing us to see, this is applied to our developing human cortex samples. This is just for one gene. This is a schizophrenia gene. Bottom line is that we identify many novel transcribed regions, new exons, alternatively spliced combinations of exons. And this has real implications, I think, for some of the genetic studies, because there are regions of the genome that are maybe being sequenced as in exome sequencing, for instance, that aren't covered in those panels. And certainly what we're hoping to do is refine functional regions of the genome for annotation in whole genome sequencing burden testing. It just highlights the sort of different types of alternative splicing marks that you can pick up. Particularly interesting to me are these sort of fusion transcripts where you see genetic material from two neighboring genes expressed in the same transcript. And we're now putting together these transcript maps together with our epigenetic profiles on the same samples to highlight how variable methylation, particularly in intragenic regions, seems to be affecting the inclusion of different exons. And just to say that many of the genes, this is important for understanding human disease. This is just a selection of genes that have been strongly implicated in dementia, Alzheimer's disease, Parkinson's disease, and motor neuron disease. In blue are known transcripts, and in red are novel transcripts that we've identified in our studies of the human cortex. It should be said that many of these transcripts are very rare, and we're still trying to work out the functional relevance of them, but the fact that they're expressed at all tells us that there is potentially novel regions of the coding genome. And just to highlight why that's important, well, we've looked at the conservation of these novel predicted coding sequences, showed that they're very highly conserved, and this kind of really suggests that there may well be functionally important sequences that could harbour pathogenic variants and are there to potentially be identified in whole genome sequencing studies. And we're currently working with cohorts such as the 100,000 Genomes Project to utilize these annotations to hopefully identify diagnostic variants in cases where these have not been previously identified. So I think I've given you a positive spin on doing epigenomics and looking at gene regulation in the human brain and in complex disease in general. I just want to put a few caveats there, and I've touched on some of those as I've gone. And that is that doing genetics is relatively easy, because it doesn't matter, when you do the sequencing, it doesn't matter what you do with your life, it doesn't matter what you expose yourselves to, it doesn't matter what cell type you do your DNA sequencing on. The disease itself doesn't affect your DNA sequence, and of course there are well annotated reference genomes. And it's worth noting that's not necessarily true for doing epigenetic epidemiology, or actually molecular epidemiology in general. And I want to highlight how that can be really important and how you can go down the wrong alley. So this is a study that we did in blood of schizophrenia patients. So one of the limitations of our brain work is it's very hard to get hold of brain samples. I really welcome some volunteers if people want to donate brains to samples, but we can't just go to the clinic and ask for a brain tissue. So we're often reliant on blood samples to get to the sort of numbers that we might need for very well-powered studies. So this is an epigenome-wide study of schizophrenia blood, and you can see that we've found some nice-looking genes, and you can start telling biological stories about how these genes might be relevant for schizophrenia. Well, that Manhattan plot is exactly the same as this Manhattan plot, and this happens to be a Manhattan plot of smoking. And actually what you may not know is that a lot of individuals, patients with schizophrenia, smoke at very high levels to self-medicate. In fact, the smoking rates are up to 90%. And so what we hadn't done was a study of schizophrenia, we'd actually done a study of smoking. So it's really worth being wary of the fact that doing this type of work, and it could be in any tissue actually, or any context, it's really important that the sort of study designs that are being used for genetic epidemiology aren't necessarily relevant for this type of work. But there are opportunities here. So the fact that smoking, for instance, has a really powerful effect on the epigenome means that I could take a DNA sample from anyone in this room and pretty much tell you very reliably whether you are a smoker or not, and how much you smoked and how long you've been smoking for. In fact, these estimates, these smoking scores that we can generate are more sensitive than just asking people through questionnaires. And of course we can then kind of use similar approaches to work out other exposure measures from individuals that might be very useful information in understanding disease, but they're also very useful to add in as confounders in these blood-based studies that will allow us to get to the very large numbers that we need. And so we're now able to actually undertake some very large epigenome-wide association studies using peripheral blood, with obviously the goal of not looking at mechanism, but potentially identifying biomarkers. This is a study of ALS that was done in almost 10,000 individuals last year, and a recent study of about 5,000 individuals with schizophrenia. So I think this is a really growing area where we can learn potentially something about disease, but certainly about the molecular signatures detectable in the blood that might relate to disease. So I hope that's clear, that we can hopefully pick up these biomarkers of disease. Another approach that I think is very useful here and a potential interest to the future is the ability to use those cell type specific marks that I mentioned earlier, the ability to pick up specific signatures of neurons, for instance. When a neuron dies, that cell-free DNA that's released from the dying cell can pass the blood-brain barrier and be passed into the blood. And so we're now sort of trying to develop methods that allow us to pick up the markers of the epigenetic signatures of neurons in plasma samples from patients that will hopefully allow us to diagnose those patients much earlier in the course of their disease and potentially allow much more effective use of disease-modifying therapies as we go on. So I just want to finish by acknowledging the group and the funders. And as I've got 3 minutes left, I also just wanted to take this opportunity just to introduce to the audience here and raise your awareness of a new initiative, the UK Human Functional Genomics Initiative, which is really focused, very much aligned to the talks you've heard today, about characterising the functional consequences of disease-associated genetic variation. It's supported by the NRC and the BBSRC. There's also support from pharma, particularly AstraZeneca, who have helped co-establish a functional genomics screening lab at the Milner Institute in Cambridge. Just want to say that we want this initiative to be a really open and collaborative network, and it's going to span both academia and industry, and there's lots of ways that everyone can get involved. So if you're interested, there is a stand here at the festival. You can scan this QR code to be pointed towards more info. And I think our website went live today, actually, so you can also check out our website to learn about the initiative and ways you can get involved and potentially get funding to do this type of work in the future. I will stop talking now and answer any questions.